What I would like to do is first to try to provide you an overview of where we are in terms of clinical trials of cardiac cell therapy in patients with chronic heart failure. I will not talk about patients with acute MI, it's a different setting. Then, as Jay has mentioned, you will have the 1-year data, the full set of data of this embryonic stem cell trial that we completed now 8 months ago. And I will finish by, because this is the title which was allocated to me, I will finish by sharing with you the kind of information we have collected from this trial and how now we try to pursue and the direction that we are now following. Now, currently there are approximately 800 cell therapy trials which have been registered, among which approximately 10% have to do with cardiovascular disease. As Jay has mentioned, the field started with the use of autologous skeletal myoblasts for different reasons that I will not repeat here. And I just would like here to share with you 2 slides. The first is the picture of this gentleman, because I think we should pay credit to this man for having accepted to be the first person in the world to receive stem cells in his heart. That was June 15, 2000, and he did very well and ultimately died from stroke long thereafter. Now, the second information has already been disclosed by Jay. It turned out that at the very beginning of this experience, the 4th patient I did surgery on did well for 15 years. Not necessarily because of the cells, because possibly of the cells, because of the concomitant coronary artery surgery. Anyway, it did well for 15 years until it deteriorated and went into severe heart failure, leading us to a conventional heart transplantation less than 1 year ago. And whereas I had absolutely no hope that the pathologist would find something in his heart, it turned out that in the scar tissue he found skeletal myotubes, which had been engrafted 15 years before, which I think is interesting, even though, once again, I would not say that the patient did well because of these cells. But as you can see, they are typical myotubes, and interestingly, some of the fibers express the slow type myosin, suggestive maybe of some form of the so-called milieu-induced differentiation, even though obviously they had not the phenotype of true cardiac cells. Now, then the field continued with bone marrow cells, and to make a long story short, currently, broadly speaking, the only cells from the bone marrow which are widely used clinically are the mesenchymal stem cells, which feature several advantages, no question about that. And if you cross the cell types with the clinical pathology, you see that actually most of the clinical trials in cardiac cell therapy right now have to do with mesenchymal stem cells, and at least 23 studies were registered on this official website not long ago. Now, interestingly, the mesenchymal stem cells are very diverse. It's a little bit like a salad. That is, you can change the dressing. And indeed, at least there are 4 dressings. There are the unmodified MACs, which have been tested, and this is probably one of the most well-designed and well-executed trial, definitely showing a benefit, as you can see, in terms of recovery of function, even though it's a relatively small sample size, with a dose-dependent functional efficacy and a correlation with an improvement in exercise testing. So this is a positive trial, no question about that. Then you have the engineered MSCs. The idea was to take bone marrow MSCs from the patient and then to engineer them so as to make them overexpressing cardiac transcription factors. So they're not cardiac cells, they're cardiac-like cells. They call them the cardiopoietic MSCs. And this group led a phase 1 trial, which was quite encouraging, leading to a phase 2. And unfortunately, the phase 2 failed to meet its primary objective, even though there was an encouraging signal in patients with the most severely dilated ventricles. But on the basis of the primary endpoint, the Chart-1 trial unfortunately failed. Then you have the enriched MSCs, and this is the X-Milocell trial, which is a combination of MSCs and macrophages, as you can see. And these investigators have reported spectacular improvements in clinical outcomes with a marked reduction in cardiac events in the treated group. Surprisingly, it did not translate into any change in the structural parameters. So there is a sort of discrepancy here, which is not that easy to understand. And finally, we have those MSCs selected on the basis of the STRO3 marker, and this is the big trial which is currently on the way, and I know that you are participating in the recruitment of these patients, so hopefully the study will turn out to be positive. So you see a lot of different trials with MSCs which are not always the same from one trial to the other, which obviously makes comparisons difficult. But then some people started to realize that maybe to repair a heart, it was better to bring and to supply it with cardiac cells or cardiac-committed cells, and to try to bring cells which phenotypically would be as close as possible to those that they are supposed to replace or to rescue. And actually we have, among adult sources, 2 types of cells that have been tested so far. We have the cardiospheres. Cardiospheres are taken by endomyocardial biopsies, exactly as we do for monitoring rejection in transplant patients. So you remove a chunk of right ventricular muscle, and then these cells aggregate and form spheres, cardiospheres, and then you collect the cells which originate from this outgrowth. Well, there was an encouraging phase 1 trial, once again, and for the youngest people in the room, I would like to draw your attention to the fact that we should be extremely careful about the interpretation of phase 1 trial regarding efficacy, because disillusions are frequent. In this particular case, the phase 1 was positive, and so they embarked into this phase 2, the Old Star, which unfortunately was stopped after an interim analysis had concluded to the probability of fertility. No probability that the study would not meet its primary endpoint, with an immediate financial consequence, as you can see, for the company trying to commercialize the cardiospheres. In the setting of adult sources, the second cell type, cardiac-committed cell type, is the cardiac stem cell. Those ones are taken during an open heart procedure from the right appendage. They're grown in culture, as you can see here. And again, there has been a phase 1 trial leading to, say, encouraging signals, the Scipio trial. Unfortunately, there are serious concerns about the data collected during this trial, and it even led to a big problem between the regulators and the institution and the center. Nevertheless, the c-KIT positive cells are still being tested in another context in addition to mesenchymal stem cells. It's the CONCERT trial, which is currently on the way. Now, our approach has been different. That is, instead of taking adult sources, we started upstream from pluripotent stem cells, iPS or ESC, in this case embryonic stem cells, because embryonic stem cells obviously can be differentiated into any lineage, in particular the cardiac one. These cells can become cardiac cells. And what is really unique with the pluripotent stem cells is not only, and I think this is important, not only that you can differentiate them down the cardiac pathway, but also they provide you with the unique possibility of controlling the stage of differentiation at which you want to collect them. And this is exactly what we have done by picking progenitor cells, but might have been as effective to take more mature cardiomyocytes. But the interesting thing with the pluripotent stem cells is this flexibility in picking really the stage of differentiation you're interested in. And just to let you know that these cells start to be tested clinically in different settings. Here, this is chronologically the first trial which has been done in humans with ES-derived oligodendrocytes in patients with spinal cord injury, with, once again, encouraging efficacy results from a limited phase 1 trial. You have another, these are the results. You have another study on macular degeneration, which is currently ongoing, so ES-derived retinal epithelial cells. You have another study starting right now with ES-derived insulin-producing cells for patients with diabetes. And very soon in China, a study using ES-derived neural progenitors for patients with Parkinson's disease. And to complete the picture with the pluripotent stem cells, you have currently in Japan a trial with iPS-derived retinal progenitor cells, again for patients with macular degeneration. And to summarize this comparison between the cardiac cells and the non-cardiac cells, you see on this meta-analysis that even though the difference failed to reach statistical significance, you have a definite trend for better outcomes when you look at the cardiac-committed cells versus those of a non-cardiac lineage, which to some extent makes sense. Now, what about now the ESCORT program? So, with the premise that we wanted to use pluripotent stem cells and differentiate them into progenitor cells, I may not have time to explain why we picked the progenitors, but if you have questions, I'll be more than happy to answer. I first would like to briefly describe the protocol, which entailed 3 major steps. The first, obviously, was the generation of the progenitors. So we started from undifferentiated, pluripotent embryonic stem cells by a phase of scale-up. And we needed, unfortunately, but we needed feeder cells, which turned out to be clinical-grade human fibroblasts. And you see here how the cells look like after an initial period of scale-up and differentiation. And so we could set up a bank, a master and working cell bank. Just to give you an idea of the scale, we started from several hundreds cells, and the bank now harbors 600 million cells. Then obviously came the cardiac specification, simple procedure. I was very keen to keep the process as straightforward and simple and cost-effective as possible. So 4 days of exposure to 2 growth factors, including BMP2, a major interest of BMP2 being that it's a drug approved for human use in orthopedics, but it's approved anyway. And doing that, you see that you can upregulate most of the cardiac transcription factors. Obviously then, you only have 50% of the cells responding to the cues, meaning that you have to discard the remaining 50%. So we selected SSAA1 as a surface marker indicating the loss of pluripotency, meaning that cells that are positive for SSAA1 are no longer pluripotent. And so you're on the safe side. And so we use immunomagnetic sorting, exactly like they do in hematology, to enrich the population in SSAA1 positive cells. And these cells, as you can see, do not express the pluripotency genes in contrast to their mother cells, which are by definition pluripotent, and conversely, they do express mesodermal cardiac genes, indicating that they have really gone into the direction you were interested in. These are the cells immediately after sorting, and you can see that their major fingerprint is the expression of islet-1, which is a marker for both the primary and the secondary art fields, meaning that these are young progenitors, very young, with the possibility of differentiating in both cardiac and vascular cells. They're cardiovascular progenitors. And this shows you that the different surface markers that have been described for identifying cardiac stem cells, and you see that the islet-1 SSAA1 markers are among those which have been described so far with others. And obviously it depends on the culture conditions and depends on the markers that you use. And so, as I told you previously, the interest is that with embryonic stem cells, that you can really pick the stage you're interested in. And we were interested in early progenitors, and we could generate them with BMP2. But now, if you want to have mature beating cardiomyocytes, well, you can do that with the so-called GY protocol based on activation inhibition of the Wnt pathway. So this is really one major interest with these cells, the flexibility they offer regarding the stage of differentiation. So these progenitors had to be tested, first for virology, the cells, the antibody, and so on, and there was no contamination. Obviously, it was critically important to ensure they remain cytogenetically stable, because as you know, when you culture these kind of cells, they go through different rounds of scale-up and differentiation and purification, and they may incur genetic abnormalities. So we use a combination of techniques. The karyotype was normal. Then we used FISH, centered on chromosomes known to be unstable during culture. And we finally used CGH arrays. And the good news was that at the end of the day, there was no genetic abnormalities in the final population. Now, oncology is obviously, tumor is obviously a critical problem. I'll come back on that in a few minutes about the clinical data themselves. But I told you that we select the cells on the basis of SSAA1 positivity. And after having discussed with the regulatory agency, we came to the conclusion that the purity had to be at least 95%. 95% positive cells for SSAA1. We're not as lucky as our colleagues in ophthalmology, because for ophthalmology, all the cells differentiate in retinal pigmental cells, so they don't have to do any selection. But we're not as lucky. We do have a selection procedure to go through. So if you take 95% of cells positive for SSAA1, obviously you have 5% of the cells which are SSAA1 negative. And the question is, can these cells be tumorigenic? So the answer, fortunately, is no. But to demonstrate that, we had to go through a series of spiking experiments in immunodeficient mice, showing that even very huge doses of this negative contaminating population failed to be tumorogenic. Actually, these cells express a mix of genes, but they were not able to cause teratomas. This is just to give you an idea of the number of viral tests that needed to be done to get clearance for the human trial on the bank, on the cells, on the antibodies, on everything. Then I was not very happy with my early experience with intramyocardial injections for different reasons. And taking advantage of the fact that as a cardiac surgeon I have the heart in front of me, we decided to switch to the epicardial delivery of a patch. And for different reasons, selected fibrin as the material of the scaffold. So again, that was a fairly straightforward procedure with a first mix of SSAO1 progenitors with fibrinogen, then the addition of thrombin to get a polymerization of the mix. And we use a surgical glue, except that we change the concentrations to make this preparation cell-friendly, and at the same time allow to have a patch that could be manipulated. Here you see this combination of concentration, 20 milligram fibrinogen, 4 units of thrombin, to have a network which did not strangle the cells, but at the same time had appropriate mechanical characteristics, allowing, as you see here, to grasp the patch and even to suture it to the epicardium. Interestingly, this patch had an elasticity in the range of 6 kilopascal, which approximately fits the range considered to be optimal for upregulation of mesodermal genes. And this is how the cells look like in the patch. You see they do express SSAO1, they do express islet 1, they even proliferate initially in the patch. So this is the last preclinical study we had to do with the cells prepared exactly as we would prepare them for the clinical trial. And that was a rat study, randomized, double-blind, with a 4-month follow-up and monthly echoes. What you see is that there was a slight effect of the fibrin patch itself, but obviously the effect was much greater when the patch was loaded with the progenitors. In particular, with a reduction in the volumes. Interestingly, recently, another group published a study with the same cells, the SSAO1, but with a different scaffold. And that was interesting to look at the two experiments because this is an independent group. And as you will see, they came to similar conclusions. So they took SSAO1 positive cells and grafted them on the core matrix. The core matrix is a decellularized porcine extracellular matrix, which is widely used in cardiac surgery for different types of repair. And here you see that the cells are seated, pretty well seated, on the SIS, the matrix. Then they grafted the scaffold on the epicardium of the heart, exactly as we did, and found a reduction in scar area associated with an improvement in recovery and, interestingly, greater paracrine effects in the group of SSAO1 cells that had been seated on the patch. So that's fairly consistent with our own data. And finally, we were concerned about enhancing graft survival. So what we did is to use the patient's pericardium as a natural bioreactor, because the pericardium is rich in supportive cells and growth factors, as you can see. And this is an operative picture where you see the fibrin patch loaded with the cells, which has been delivered onto the epicardium of the infarct area and which is covered by a pericardial flap. So the pericardial flap is folded over the fibrin patch. It allows to secure it in place. It's what I've called a kangaroo operation because you have a pocket. And then this pericardial flap is sutured all around So it maintains the fibrin patch in place, and hopefully it provides the patch with some trophic support intended to enhance the viability of the cellular graft, at least initially. Obviously, because there are allogenic cells, we had to give immunosuppression, but you will understand why in a few minutes. That was a short-term immunosuppression, 2 months for the first patient and then 1 month, and with low-dose regimen to avoid any drug-associated side effects. So these are the inclusion criteria of the trial, fairly standard inclusion criteria for this kind of study. Obviously, an history of infarct with a localized area to put a patch on. Obviously, severe left ventricular dysfunction, and for ethical reasons, because that was first in man, indication for coronary bypass. So this is the population, the description of the population. The first patient died, unfortunately, as often in this kind of trial, because we started by a compassionate indication, and the surgery was okay, but a patient died thereafter from multiple comorbidities, which are listed at the bottom of the slide, and that was nothing to do with the cell, so we had permission to move on. The other patients had really very smooth post-op courses, and unexpectedly smooth given the severity of their left ventricular dysfunction. The primary endpoint was really safety. Safety and again safety. And when I say safety, we're thinking in terms of tumor, once again, arrhythmias, and allosensitization, primarily. And here you see the different examinations that were used to monitor the patients for these different complications. Now, one important feature is the batch release criteria, because this is a critical moment, the critical time at which the cell is going to release the lot which is going to be put in the patient, and this is irreversible. So the big issue here, and that's why I spent just 1 minute on that, is the identity. To be sure that the cells are really cardiac committed cells expressing islet 1, but that they are really have lost their pluripotency to a certain level. And so we took NANOG as a marker of pluripotency, and both NANOG and islet 1 had to be below or above a certain threshold compared with the mother cells, that is the undifferentiated pluripotent stem cells. And that was done by PCR in triplicate by 2 independent and separate investigators to be sure that the release criteria were met and that we would not deliver a product that would not exactly match the preset criteria. Now that said, it's very tough to define these markers of pluripotency. Why? Because markers of pluripotency, the common markers, are also markers for lineage differentiation. For example, OCT4, which is commonly used as a marker for pluripotency, OCT4 is also a marker for cardiac differentiation. SOX2, which is a classical marker for pluripotency, is also a marker for neuroectodermal differentiation. So it's tricky. And on top of that, you should know that these markers are actually expressed in adult cells. These are human cardiac tissue obtained in patients from neonates and adults. And you see that these cells, some of them, do express NANOG, OCT4, and SOX2. And these markers are also present in mesenchymal stem cells and in cord blood cells, which are routinely used clinically. So we have to use them, but I wanted to point that it's not that trivial to select the good marker and the appropriate level of differentiation, because expression of the marker does not necessarily mean that the cells will give rise to a tumor. Back to the results, the black bars here indicate the preset release criteria. So you see that we were good in terms of viability as well as in terms of purity. We even went to 98% purity. And importantly, the expression of NANOG and islet-1 fully met the preset release criteria. Now, regarding the outcomes, as I told you, the first one was the assessment for tumors using PET scan and whole body CT scan at 2 different time points, and so far, so good. There has been no new abnormal proliferation either in the heart or elsewhere, and only the last patient didn't still have his 1-year follow-up because I did surgery 8 months ago, so we still have to wait 4 months for him to get his CT scan. Now, as an additional safety check, we looked at the circulating cell-derived DNA in the patient's blood in case cells would have migrated off the patch. So we worked then closely with our colleagues in oncology to use high-throughput sequencing, and we were fortunate. We were fortunate because it turned out that the cell line harbored polymorphisms which were rather unusual. And so none of our patients had this polymorphism in their DNA before surgery, which meant that if this polymorphism had appeared after the operation, that would have been suggestive, obviously, of a migration of the cells out of the patch. But the good news is that none of this polymorphism was detected in any patient postoperatively. This is just an example. You see the CT ratio in the cell line for this particular polymorphism, CT, and you see that the ratio is markedly different in this particular patient, and that was the case for all patients. Then we'll look for arrhythmias. All patients had an internal defibrillator pre-op. Some of them already had one because of the disease. Others were implanted. And so we looked at those various events, and again, so far, so good. There has been no arrhythmia so far in any patient detected by the ICDs. Now, regarding the allosensitization, this is the immunophenotyping of the population. As you can see, these progenitors do express a little bit of class 1. They do not express class 2. They do not express co-stimulatory molecules. They do express ligands for natural killer cells. Now, interestingly, when you stimulate the cells with interferon gamma, you markedly upregulate HLA-1 expression in the cells. So the reasoning is that when you graft the cells with a patch and so on, you create unavoidably some inflammation. So this is a sort of mimicking of what might occur in vivo at the time of perioperative inflammation, this upregulation of HLA-1, which to some extent is a good news because that should limit damage induced by NK cells. And as I told you, the progenitors do have some receptors for NK cells, so we hope that at least that not too many of them could have been damaged by NK cells. And indeed, when we did MLRs in this patient, we found that 4 of them had virtually no response. By MLR, I mean that you take the patient's blood and you put it in contact with the progenitors and see whether you have a proliferation of immune cells like CD4 or CD8. You see that you do not have any response in this patient, that patient, that patient. Conversely, if you take the same patient's blood and expose it to other allogenic blood, then you get the expected response. Now, we only have 2 patients with, well, let's say some response, but still much lower than the one you get with the allogenic peripheral blood mononuclear cells. So 3 patients developed donor-specific antibodies against antigens specific for the cell line. That was not really unexpected. Those people using allogenic MSCs have also reported alloimmunization. For me, good news, because it means that at least for a while the cells were there. But I would like to draw your attention to the fact that the highest mean fluorescence intensity was in the range of 4,000. And it has been shown that it's only above 5,000 that donor-specific antibodies start to be cytotoxic. So we're not so much concerned that these antibodies may have contributed to the death of the grafted cells. And that was further justification for this low-dose immunosuppressive regimen. You see here the mean doses of cyclosporin and of MMF. Indeed, we followed the level of immunosuppression by looking at the response to challenge by viral immunopeptides. And basically what this graph shows is that postoperatively the response were not significantly different from what they were before surgery. In other words, these patients were not dramatically immunosuppressed, and that's what we were looking for. Now, regarding the clinical results, I think that I should be once again very clear. It's not an efficacy study, so I can just show you the data, but obviously it would be completely stupid to draw conclusions regarding efficacy because of the small size, because it's not blinded, and because you have the concomitant bypass. So as you can see, they did well. Well, good news, they did well. Ejection fraction rose over time, which could be simply due to the revascularization. And obviously, we have been interested in looking more specifically at the segments that had been transplanted with the cells and the patch. And what you see is that there is a decrease over time of the wall motion score of the transplanted segments. And the lower the score, the better the contraction, based on the classification that you see on the top of the slide. So does it mean that the cells have improved the wall motion score? I say it loud and clear, I do not know. And I will not claim that the cells have worked. That's clear. I will not claim that they have worked. This is the result. Could be due to a lot of things, maybe to the cells, maybe to the patch, maybe to the bypass, maybe to I don't know what. I just show you three brief examples. This is patient number 2. She's now 3 years from her surgery, doing very well. She was in pretty bad condition initially. She was sitting and sleeping in her armchair to breathe. And you can see that the thickening of the wall has markedly changed over time. I see Dr. Prabhu looking at very carefully, so I'm afraid of his comments then. This is another patient, interesting patient, a large anterior infarct, completely occluded LAD. So no bypass on the LAD. The bypass was on the lateral wall. And I just keep that a few seconds. And the last one, it's more tricky because it's a large akinetic, dyskinetic area. There was a slight, but I would say very slight improvement only, if any. So, how does it work if it works? Well, you have, as you know, two major theories. The first is the so-called remuscularization. That is, we are going to inject or transplant huge doses of cells with the hope that they can make a new myocardial tissue, and that would be great. I think that there are many challenges. The first is that you really need a large number of cells if you want to repopulate these hearts. The second is that you need non-cardiac cells, because it has been shown that the technique worked better if you added some stromal cells or endothelial cells So it further complicates the procedure if you have to transplant 2 or 3 different cell populations. You obviously have to keep them alive, because here the objective is to have a new myocardial tissue which would stay there forever. So because if you use allogenic cells, at least, you need to immunosuppress them for a long period of time. And I draw your attention to the fact that in this ES-derived retinal pigment cells for macular degeneration, the only complication that has been reported has to do with immunosuppression. No tumor, but immunosuppression-related side effects. So if you graft huge doses of allogenic cells and want them to stay forever, you may get into trouble with side effects of immunosuppression in this very sick patient population. And finally, and this is a real challenge, if you graft the cells, you have them to couple, and to couple appropriately with the host cardiomyocytes, otherwise you may get into troubles with arrhythmias. So the other concept is based on paracrine signaling, assuming that the cells are actually acting as biofactories, releasing factors that can harness endogenous reaper pathways, and that after a while they will disappear. There are several lines of evidence for that. Probably the most compelling evidence is the discrepancy between the disappearance of the cells and the maintenance of a functional benefit. This is one example among others. You see here an improvement in function after 4 months, a decrease in infarct size. These are in this particular bone marrow multipotent progenitor cells. And once again, at 4 months, you have a definite functional improvement. But at the same time point, there is not a single cell remaining on board. So you have to explain that. And interestingly, at the same time point, you have marked changes in the gene profile of the recipient hearts, suggesting that the grafted cells have released factors that have changed the genome of the recipient myocardium. Now, the second evidence is that it has been shown that you did not necessarily need electromechanical integration for the improvement to occur. And finally, we know that cells release a lot of factors. And so if you look at this complex picture of the pathophysiology of heart failure, it has been reported that actually many of them can be appropriately addressed by the factors released by stem cells. So, increasingly, it is recognized that at least part of the factors are clustered in extracellular vesicles. Extracellular vesicles released by all cells in the body. And I use the term extracellular vesicles because it's an heterogeneous entity with exosomes, but also microparticles and apoptotic bodies. And these are super shuttles. These are really super shuttles that transfer, in particular, microRNAs, but also proteins and lipids to recipient cells, and they can change the genotype and the phenotype of the recipient cells. So the first study we did, at the time I was transplanting the patients clinically, is to assess the effects of the extracellular vesicles derived from these progenitors, exactly those that I was transplanting clinically. So what we did is to take the SSA1 progenitors, stress them to increase the production of vesicles, and then we transplanted either we injected either the control medium or the extracellular vesicles or their mother cells. So I stress the fact that this is the same batch of progenitors, half being transplanted, half being used for collecting the vesicles. And we use this mouse model of chronic heart failure, where you induce infarction by a coronary ligation and wait for 3 weeks. And after 3 weeks, once you have a scar, you inject either the medium or the EVs or you transplant the cells. And we did that, in a substantial number of animals because of mortality and so on. Now, as you know, the issue with the mouse model is that once you've done the coronary ligation, usually you test the therapeutic product immediately, because if you have to reoperate on the animals, mortality is extremely high. So to avoid that, we use transcutaneous injections under echo guidance, and mortality is virtually zero because it's a non-invasive procedure. You see, you look at the needle going through the skin and injecting around the infarct area. And basically we found similar improvements in outcomes between the cells and the EVs derived from those cells. You see here the decrease in volumes, which are exactly similar with the progenitors and the vesicles. Then we looked at the signaling pathways in the recipient hearts and found once again that basically the same pathways had been turned on either by the cells or by the cell-derived vesicles. We took two patches similar to the ones that I transplanted in patients and look at the conditioned medium released by the cells, which were embedded into these fibrin patches, and found a predominant expression of microRNA 302, which has been shown to be involved in cardiomyocyte proliferation. And again, these are the patches which were implanted clinically. So next, we wanted to confirm this data and switch from ESLs to IPS as the source of the cardiovascular progenitors for a reason of practicality, primarily. And basically, the cardiovascular progenitors you get from the IPS are extremely similar to those you get from the ESLs. And we characterize them. These are commercially available IPS from Cellular Dynamics. And you see that they have shut down the pluripotency markers, which is good. They do express markers of a progenitor state, and they do not express markers for more major cardiomyocytes. So they're really cardiovascular progenitors as confirmed by these immunostaining studies. So we checked that these progenitors were able to release vesicles, and given the size of the released vesicles, most of them are likely to be exosomes, usually in the range of 100 nanometers, confirmed by cryon microscopy. Then the first thing to do was to really be sure that these vesicles could be internalized into recipient cells. So we use H9C2 cardiomyoblast and expose them to the vesicles from the cardiovascular progenitors. And these are control on-label EVs, so you don't see anything. This is another control, and this is what you get when you have labeled the vesicles. So really the demonstration of an internalization of the vesicles into the recipient cells. So one step further, because these vesicles shuttle microRNA and proteins, we try to figure out whether specifically these two fractions were internalized, and we use a specific dye for proteins, and you can see that there is evidence for internalization of the protein content of the vesicles into the recipient cells. And then we use another dye specific for RNA, and the RNA cargo was also shown to be internalized. Then we try to figure out in a more dynamic fashion whether this event really happened. Then we design potency assays to ensure that these vesicles were doing something in vitro. So it's a fairly standard model. Again, we find our H9C2, and this is the control where the cells are allowed to proliferate then you deprive them from serum, and you get a marked decrease in viability. And then this is another control with fetal bovine serum EVs. And then we added our vesicles, and you can see this dose-dependent improvement in viability. And I can tell you that we have done that at least now 40 times, and it's perfectly reproducible. So you really have a dose-dependent improvement in the viability of stressed cells when you expose them to the vesicles. As an additional confirmation, we use another potency assay, which is an angiogenesis assay using UVEC as the recipient cells, because the previous cells you have seen that were rat cells. So we wanted to check whether it also worked with human cells. And again, you see that you have a dose-dependent improvement, increase in angiogenesis in the scratch test. What is the scratch test? You take human umbilical vein cells, and once they are confluent with a micropipette, you scratch, you make a wound, and then you monitor the migration of the endothelial cells to heal the wound. And in a complete media, you see that you have a full recovery, full healing. This is a negative control, that is a poor media without the growth factors required for the umbilical cells to migrate, and you don't have anything, you don't have healing. And this is the same poor media with the vesicles, and you see that you have a partial recovery, a partial healing of the wound. And finally, the last assay we did in vitro was to monitor whether they could do something on cardiomyocyte proliferation. So this is a line, ES-derived line, ES-derived cardiomyocyte line, and exposed to vesicles once again. And you see that there was, again, a dose-dependent improvement in the proliferation of these cardiomyocytes, further documented by immunostainings. Now, we then looked at the content of the vesicles, in particular the content in microRNA. And what you see here is that as you are increasing the threshold, you have more microRNAs in the vesicles than in their parent cells, meaning that you seem to have a selective packaging. It's not a copy and paste. It's not, random distribution. It's really a selective packaging of the vesicles in the exosomes. We found that 16 of them were overexpressed, taking microRNAs broadly expressed across different species, and most of them were actually oriented towards tissue repair, as you can see here. And the mechanistic question then was, can we make a relationship between the expression of these microRNAs in the vesicles and the effects that we see in terms of viability and so on? So to try to answer this question, we took 3 different preparations of vesicles. We exposed again the rat cardiomyoblasts to these vesicles, and then we screened the genome and looked at the genes which were upregulated or downregulated in the cells that had not been exposed to vesicles versus those which had been exposed to vesicles. And we found that among the genes that were downregulated, some of them were possible targets for some of the microRNAs isolated in the vesicles. In other words, that tends to indicate a mechanistic link between some of the microRNAs present in the vesicles and the effects that are seen with regard to viability. And this is just one example of the kind of picture that you get with one microRNA and one signaling pathway. So then we need to validate that in vivo, so we use the same model. and compare control medium with iPS-derived cardiomyocyte, mature cardiomyocytes, versus progenitor-derived EVs. Now, you can ask the question, why did you not test EVs from cardiomyocyte? The answer is very simple. Mature cardiomyocyte release very few vesicles, very few. The younger the cells, the greater the production of vesicles. So that's the reason. And what we found is that actually the best outcomes were seen. That was again randomized and blinded study. And double-blind. Double-blind, that is blinded regarding echo collection and blinding regarding statistical assessment. We found that the best outcome were seen in the vesicle group. So if you compare with the progenitors, you get the impression that they did better. Actually, they did not do really better. The reason for the result is that if you stratify the animals between the responders and the non-responders, the interesting observation is that you have a greater scattering of data in the cell group compared with the vesicle group. And that makes sense, and that's consistent with what has been reported in clinical trial, that with the cells, you may have cells clumped in the syringe, you may have cells dying more or less rapidly following the injections, whereas with a suspension, just like a drug, you have probably a more homogeneous distribution of the product when you inject it. So, you know, taking left ventricular and systolic volume as the primary endpoint, so less 5% of response defines the non-responders, and this is the improvement in the responder group. And again, you see this marked distribution, scattering of the data in the cell group, whereas the data are much more grouped together in the vesicle group. We also looked at the pathways that had been turned on in the recipient hearts, and to make a long story short, found that again in the vesicle group we found upregulation of pathways going, I would say, in the right direction of cardioprotection, tissue repair, proliferation, survival, and so on. So I don't know whether the vesicles are a miraculous treatment. I don't think so. But definitely they offer significant advantages if you're thinking in terms of widespread clinical applications listed here. And in particular, the fact that they can be frozen without losing their efficacy. They can be stored and available off the shelf. They can be easily injectable. And manufacturing, quality controls, and reproducibility are likely easier and more consistent than what you can get with purely cellular products. Finally, interestingly, our preliminary data show that the vesicles do not seem to be immunogenic. You see, again, 3 different preparations. I stress the fact that I want every time different preparations at different days and different weeks to be sure that the data are reproducible. And each preparation then is done in duplicate or triplicate. Okay? And what you can see is that they do not trigger a CD4 or CD8 proliferation. They do not seem to induce NK degranulations, but they are not immunomodulatory. That is, if you use PHA to induce an immune response, you will not ablate this immune response by the vesicles. At least that's what we have found so far. I want to finish by showing that this equivalence that I have shown to you between cells and cell-derived vesicles has been found in completely different settings, which is interesting because this data come from independent labs working on different diseases. This is a study on neuroprotection, and what you can see here, they have 2 different preparations of MSCs. They have 2 different preparations of EVs derived from these MSCs, and basically they found exactly similar improvements in outcomes regardless of whether they use the MSCs or the vesicles that they have secreted. This is another example. This is a retinal injury, and I just draw your attention on the blue arrows showing that, again, MACs and exosomes from MACs, no matter the endpoint, you need to be an ophthalmologist to understand that, but you see that you get similar outcomes compared with control with both the cells and the vesicles. And I think that looking at what happened in other areas than cardiology is always instructive, because once again, these are three different labs, the ophthalmologist, the brain people, and our lab, basically finding the same thing. That is, that you can recapitulate most of the effects of the cells by the sole administration of the vesicles that secrete. So if we go back to this sketch depicting the evolution of the use of cells over time, it might be that the next page could be the page of the paracrine factors, either the vesicles or a fraction of the vesicles, or maybe some components of the soup that we currently use. But that's at least, this is, at least is a path, I think, which is worth being explored. And if I finish by this a very critical editorial recently published in Nature, it's interesting that they stress the use of cardiomyocyte or progenitors, that is cardiac commuted cells, but one step further one could think of using these cells no longer as the agents to be transplanted, but as the factories remaining ex vivo for producing the factors which would be the only therapeutics to be delivered to the patients. That said, I'd like to obviously thank you for your patience, acknowledge really the contribution of my colleagues who have made all that possible, and show you the picture of the EV Dream team extensively working now on this project. Thank you very much.
Thank you, Dr. Monachie. This is very exciting unpublished data. So I guess you are planning to the next clinical trial is vesicle therapy.
Yes, we're in the process of doing the translational phase. We still have much work ahead, in particular for purification step. We are going to
Do we have time for a few questions?
Okay, first, the human study is very difficult because it's difficult to get the control. Okay, generate the control group. You have shown your patient after several months, the cardiac function improved, but that's the same you compare to the probe. So one of the possible variables is maybe you took care of the patient too well. So without any treatment, the cardiac function will improve. So in your S-core study, what is your control?
Well, maybe I've not been clear enough. The S-core study was the first in-man trial, and its exclusive endpoint was safety.
Yes,
so that's why I have said that you cannot draw. I completely agree with you. You cannot draw any meaningful conclusion regarding the so-called efficacy.
But you have shown the data.
Yeah, I showed the data. At least it means that the cells have not been harmful. But I will not claim, I think I've said it clearly, but I say it again. I cannot say that the improvement was due to the cells or to the
But in the future,
sure,
if you reach the phase
absolutely.
3, what will be the control?
Well, the control, first of all, the next phase will be for us the vesicles. But if we had to go on with the cells, the control, for example, would be a fibrin patch without the cells, or nothing. But the fibrin patch, as we did in the animal experiments, the cell-free patch could be a control.
GQ.
It's a wonderful talk. I appreciate it.
Thank you.
And I have just two small questions about the use vesicles from the stem cells. So it's a proper control for the microvesicle from given the cell type as versus progenitor cells.
Yeah, you're right. It has been done. We haven't done that, but it has been done, and clearly there is a cell specificity. In other words, if you take vesicles from fibroblasts, for example, it doesn't work. Now, maybe vesicles from MSCs could work as well, although unpublished data from an Italian group which has extensively worked in that has shown that the efficacy of MSC-derived EVs were not as good as the efficacy of EVs taken from cardiac stem cells. They have not published that yet, but I mean, we should be open-minded, and maybe MSCs-derived EVs could do the job, but at least fibroblast, it doesn't work.
Rosama?
Very interesting pieces of studies there. So two questions. One of the question is, now we, at the end of the presentation, now I can conclude that the effect was due to exosomes, right? And you at some point showed there is increase in microRNA 206 in the exosomes, and that is one of the beneficial mechanisms that you are proposing. Is it possible to check the serum in these patients if there is a increase in this particular microRNA? Because if we believe that it is because of the secretion, probably it should be circulating in the patient serum as well, serum or plasma. So if you could check that, I don't know if we can expect that microRNA could be.
I don't know whether right now we could do that. I also wonder whether the amounts of this microRNA which would possibly circulate would be enough to be detectable by the methods that are currently used. But to be honest, the data that you're alluding to, I mean, the release of the microRNA from the patch were done almost at the end of the clinical trial, so we had not anticipated that. And as you know, once the protocol has been defined and all the blood samples have been programmed and validated by the ethics committee, it's a little bit difficult then to change everything. So I'm not sure that we would be able to do that. Now that we know that, if we had known that before, but I'm afraid it's a little late now.
Well, time is running late.
Final question.
Short question. Short question.
Yes. In your ESCOT trial, you showed that this ES-derived cardiomyocytes, it does not contribute to any kind of malignancy. Are you aware of any other clinical trials where the ES cell or the iPS cells transplantation led to any kind of malignancy or metastasis? For example, whether inside the heart, whether it created a kind of embryonic carcinoma, or whether it is metastasized to other organs and created. Are you aware of any other clinical trials where it is not safe? Any of these two?
So far, and with the caveat that you don't have hundreds of patients, there has been no report in any of the ES trial of malignancy. And the only real complication, as I mentioned, and this is important to keep in mind for us with cardiac patient, was complications from immunosuppression. Now with IPS, when they started the trial in Japan, for the second patient, they found possibly oncogenic mutations in the line and did not transplant the cells, stopped the trial, and that they have moved again, but with allogenic cells. That was the only example, but that was a possible oncogenic mutation in the IPS, possibly due to the reprogramming process. But for patients having received ES-derived differentiated, more or less differentiated cells, so far so good.
Is it because
all right, you can talk maybe tomorrow or afterwards, but the time is running late. Let's appreciate Dr.